Very good morning to all my teachers here and all of you. It's an honor to be called here for the second Dr. Agarwal Memorial lecture, and I thank my teacher Dr. Shubha for inviting me. So today this topic also has been nicely suggested by her as ATGC, every letter accounts G. So I will try and justify why every letter, these 4 letters, make what we are or make whatever we remain throughout our life. So let me begin by paying homage to, I should say, my grand teacher, because he was never my teacher, he was my teacher's teacher. I didn't really see him as a teacher, but I was fortunate enough, like he was my examiner in my DM exam, but I was fortunate to have him in our RAPSAC membership in CDFD. So the initial years when I joined CDFD in 2007, every year he would have some of his ideas, as madam told, he would give unrealistic aims and he would say, you do this, and at that time it looked like totally impossible. And I'm really fortunate that when I started my laboratory in Center for DNA Fingerprinting and Diagnostics about 10 years back, the objectives of my laboratory, which I was thinking what to put, and they were actually dictated to me by Dr. Agarwal, and I am still trying to achieve those objectives, and it's a forever reminder for me every year whether I have achieved them and what I need to do in future. He is really a father figure for all medical geneticists in India, and we would never be there as a specialty without his efforts. So with that remembrance of Dr. Agarwal, let me go on to my talk. So basically I have tried to put into some stories about this ATGC. We saw a nice skit now, and I will try to tell you my interpretation of what these small letters mean to us. So let's begin with the letter A. The letter A, I have thought it as a book of life, because if you look at DNA, it is also a book of life that tells how our life needs to go. And so if you look at any book that we read, so most of our books that we read are all written using alphabets A to Z. So in English language, we use 26 letters to write all the books that we can create. But our DNA again uses nucleotides as alphabets and writes all the codes for all the proteins that are needed in our own body just using the 4 letters A, T, G, and C in different proportions. So it's much advanced, much more advanced language compared to the English language that we use. So using these 4 letters, the DNA makes all the proteins required for formation of a human being. And as we can write multiple different types of books using the letters, in the same way, DNA writes using just these 4 letters and creates different human beings throughout our world in very different ways. So it is just the difference of DNA between which makes us different from each other. However, as we have seen just now in the skit, just like we make mistakes while writing, similarly the DNA also makes some mistake when it has to replicate itself, and that is what we call as mutation, when a particular nucleotide is different, but it creates havoc in our body, it leads to diseases. So the typical example to understand is of sickle cell anemia, a very common disorder where just this A changes to T, which leads to just change of one amino acid, and that leads to gross abnormality in the red blood cells of the body and a severe disease of sickle cell anemia. So just a small change, of one letter change in a 3 billion base pairs leads to a devastating disease. That is the power, or that is the importance of DNA sequence. And we as geneticists, our main job is to find these mistakes which the DNA creates, so that these mistakes either can be treated, either can be corrected, or we can do something about them. Now finding these mistakes, it is much easy if you know where a mistake is. So if you want to see that this is a book and you want to find mistake here, now if this book is published by someone else, if we know where is the mistake, we can easily go there and see is the mistake there or not there, and then say whether the mutation is there or not there. And that is what we have been doing throughout, that we just try to sequence the gene and we try to see whether there is a G or a T at the particular place. This is very easy if we know what to find. But what is going to happen if we don't know what to find? And that is where the problem comes. So if we know sickle cell anemia, throughout the world it is caused due to one mutation. So any patient with sickle cell anemia, we can directly go to that position and look whether there is a or a T. But if a child comes with intellectual disability, or if a child comes with some other problem, we don't know where to look for, and there if you have to find mutation, it is like finding this dot in the Encyclopedia Britannica. That is what is human genome, 3 billion base pairs, and you want to find one mutation. And if you see, it is something like finding needle in a haystack. So if you have to find this small needle in this haystack, can think what would be the complexities. And about 4 years or 5 years back, we would have never thought of doing this. But over the last 4 or 5 years, the technologies which have come, called as next generation sequencing strategies, using different platforms, now it is actually possible to sequence this whole DNA and try to find what is the problem with this. But again, as I said, instead of sequencing the whole genome, which is like sequencing all these books, all the books, instead of that, we could just take out one important book, because the exome or the coding region, it forms only 1% of the human genome, but more than 85 to 90% of mutations are in the coding region. So instead of sequencing the whole genome, we could just look at the exome, and that way we could identify most of the diseases, and that is what is practically done. And you can see what is the problem here. If we sequence whole genome, you are going to get about 22 lakh variants, as opposed to that about 22,000 variants in exome. So from analysis point of view and from yield point of view, it is always good to do a exome sequencing compared to a whole genome sequencing. So mostly in our clinical practice, we try to sequence the exome and try to find what is the cause of the disease. But as we said, finding needle in a haystack, because sequencing has now become easy, but sequencing doesn't mean anything. Printing a book doesn't mean anything. Finding the mistake in that is something which is difficult, and that is where the expertise of either a bioinformatician or a clinician, whoever is involved in this exome sequencing, comes into it. And so finding needle in a haystack, the strategy, should it be something like this? Are you going to find the needle if you do something like this? Very difficult to find a needle in this way. Rather than that, what we would do is that we assemble a haystack, maybe then assemble it into small packs, then take one pack at a time, and maybe you will find different needles in that pack. So you will have to put some sieve, remove the unwanted needles, and then reach the needle. And exactly this is what we do in exome sequencing. What we call it is the next generation sequencing pipeline. And here, what is the drastic change is happening is that the new age doctor or the new age medical geneticist has to have friendship with the computer. It is not manually possible to do a exome sequencing. A Sanger sequencing can be manually interpreted. You just have to see ATGC, nice figures come, but the data which comes out of exome sequencing is impossible to be analyzed manually. And we have to make friendship with our friend computer here. We need to become computer literate. We need to know to analyze big data, and that is very important for the new age clinician. However, you can think of how much is the data deluge, what data is generated. If you see a typical exome, our exome is about 50 MB, and you do a 100x coverage. Each exome sequencing generates about 5 GB of data. Can you imagine? 5 GB used to be the whole memory of a computer in some days before. And then when you sequence this whole genome, you find the variants. You find about 25,000 variants in each individual. Now out of this 25,000, you have to find one. That is the issue involved. And so these data issues are there still, but we have to tackle them. Mainly the data issues involved are in analysis and in storage. High power computing is needed. A normal computer cannot do this kind of analysis. In the initial days when we started doing exome sequencing, we really did not have the high power computers. And so we tried doing mapping on our routine laptop with a 4 GB RAM, and with that RAM, the first exome that we tried to map, for 1 month it was running day and night, and still it could not map. And that is when we had to go for high power computing. The computer that we use for analysis now is having a RAM of 128 GB. Can you imagine 1 GB or 2 GB is the desktop RAM that we have in most of our computers. But this kind of computing needs such a high degree of analysis power as well as storage. You can imagine each exome is 5 GB and the files it creates is another 5-6 GB. 10 GB of sequence for one patient, and if you do 10 patients, 100 patients, where are you going to store this whole information? So these all issues are coming up, and now to think even of doing whole genome is going to, one patient is going to give you 90 GB of data. So it's really beyond our thinking, but still it is what we have to do in future. It is the future of medicine. We have to get adjusted to this kind of thing. But with this whole sequencing, now we really have the power to understand our genome, to understand the diseases better, to understand and develop new treatments. But at the same time, as Dr. Shubha said, as Spider-Man told us, with great power comes great responsibility. Hence, with so much of power of having, we can know the past, present, future of that particular patient, and when we know all that, it is very important that we use this power with responsibility, and that brings us to a lot of ethical, legal, social issues that are coming up with this kind of sequencing. So cost is a major issue still now. Maybe we can get a exome sequencing done for about 50 or 60,000 rupees, may not be still affordable for many of the patients. But more importantly, you tend to find incidental findings. You tend to find finding something which you are not looking at. See, when you are doing sequencing for thalassemia or sickle cell anemia, you are only looking for that gene. So if a patient is thalassemia, you are sequencing beta globin gene, giving the result. Here what is happening is that you did sequencing for thalassemia and you find that he is at predisposition for a medullary thyroid carcinoma, or he is at predisposition for breast cancer, BRCA1 mutation is found. Now what do? Do you tell the patient about that? Because patient did not come to you for cancer counseling, he came to you for thalassemia. Now do you tell him that or not? So ACMG or American College of Medical Genetics has made a list of about 56 variants which have to be reported because they are correctable. So here the issue comes about return of findings, whether to tell or not to tell, because ACMG decided you should tell the findings only if they are correctable. If some finding is not correctable, then telling that does not help the patient, but he is going to be under that impression that he is going to develop either predisposition to cancer or some late onset disorder, like some diseases come in late life. All genetic diseases don't come in childhood. Now whether we should tell about those disorders, because unless we can do something about it, there is no point telling the patient. So all these issues come up, and that is what we try to tackle in the pre and post test counseling is very important. Exome sequencing is not a test like a hemoglobin you just order and get the result. A pre-test counseling is very, very important, where we try to tell the patient what is this test going to give us, then what extra information is going to come, and whether the patient would like to get that information or not, and then a post-test counseling about what has come out in the result. And as we will see later on, we may not get anything at the end of exome sequencing also, because we still don't know everything about all the genes. There are 25,000 genes in humans, but only 5,000 genes are, the diseases are known. Another 20,000 we don't even know what they cause. So it is still, although we have the power to sequence, we don't have the ability to analyze it fully. So this brings to show you one experience of a patient where we did exome sequencing. And this whole story started in 1999, and the whole story started in SGPGI when Dr. Shubha had seen this case. This patient, you can see the abnormalities in the hand and the leg, and this child had the family had another child who was diagnosed in uterus by ultrasound, and the fetus was born with exactly the same abnormality. And you can see this is the black and white photo era of 1999. But we could really not do anything. Only at that time it was reported in American Journal of Medical Genetics as a new entity, and it got reported in OMIM as a new entity, and it remained there like that only. However, in 2012, when we had the capacity to do exome sequencing, Dr. Shubha contacted the patient again, and now this small child was an adult and you can see she had undergone multiple plastic surgeries, and the hands were quite functional. She was able to do her routine work, but we thought we should try and find out what was the gene which caused this problem, and over that 99 to 2012 there was no gene identified for this. So we went ahead and did exome sequencing for this family, and as I told you, this is the type of analysis that we do. So we get about 27,000 different variants in the patient. It goes through various levels of filtering. We filter out for the normal variants, we filter out for the synonymous variants. So after all that filtering, about 14 variants remained, or 14 mutations, but all mutations don't cause disease. Some mutations are polymorphisms, they are the ones which make us different. And so of this we found one particular mutation in this gene BHLH-A9, and this was the mutation, a deletion insertion of TT at this position. Now finding this mutation doesn't make that it is disease causing, because till that time it was not known what this gene does. So we went ahead and did Sanger confirmation. You can see this is GA in the normal and it is TT in the patient. So it was confirmed by Sanger sequencing, what we got in exome sequencing. But then we did not know what it does. So we went and saw in mouse model what it is doing, and what we found was that this gene is particularly expressed only in the hands and leg of the mouse, and in mouse you can see something similar to what happens in humans was seen. So it gave us more confidence that this is really the gene which is causing the particular phenotype. And then when we saw further, we saw that this particular is the phenotype in our patient, and we saw that duplication of this gene leads to another abnormality called as split hand foot malformation, and another group had reported that this particular abnormality of cynostotic syndactyly is caused due to mutations. And then we were sure enough that our patient, this gene was the responsible gene for this particular abnormality. However, it is so much different from the phenotype which is reported earlier, and we are still doing research to find out exactly why this mutation was so severe to cause such an abnormality compared to the other patients. So we could publish this article as a new gene identified for this particular entity after about 15 years of identifying the patient. And for me it was a kind of a Guru Dakshina that what madam started in 1999, I could finish it in 2015. So that was a very nice experience to begin with exome sequencing. And after that we have been doing lots of this exome sequencing, and we can see that we have a good yield from them. About 23 whole exomes, 13 were positive. About clinical exomes, 17, 6 were positive. So ongoing, this is an ongoing process where we are also improving our abilities to do this exome sequencing. Similarly, it has utility in molecular oncology to identify cancer predisposition syndromes or to identify somatic mutations so that treatment can be geared to it, or even these pathogenic organisms. Nowadays we can easily identify which organism is causing outbreak or antimicrobial resistance, because these organisms have very small genomes. Easily they can be sequenced quickly and we have the answers. So that is what we have till now, that we have a capacity to find the mutation or find the variant. But this brings us to the next letter, that is G. So finding a variant doesn't mean anything. You have to prove that that variant is the real variant. And that is what I want to tell you in the form of a Goldsmith's dilemma. So if you go to a shop and you see all these ornaments, can you tell which is real or which is having how much of gold or not? So for a goldsmith it is very important. He has to find out how much of gold is there, because he is going to give money to you in respect to that. So what will he do? He is going to manually inspect, he is going to use some keratometer to see, or at last he is going to melt that gold and then tell you how much of gold is there. So ultimately दूध का दूध पानी का पानी हो जाएगा, वो बताएगा आपके इस ornament में सिर्फ 50% gold है, इतना ही पैसा मिलेगा. So that is also the dilemma that we face always. When we find a variant, we have to identify whether it is a disease causing mutation or a polymorphism, because we are giving a brand to that patient. That patient will be labeled with that disease. So it is our responsibility to tell for sure whether it is really a mutation or a polymorphism. So for this we use various strategies to come to the end result. So I'll just take you through this one patient here. So here is a 5-year-old male who came with joint contractures, normal IQ, coarse facies, corneal clouding. So with these clinical features, we can make a clinical diagnosis that probably he is a mucopolysaccharidosis patient. We do an enzyme assay, we find that aryl sulfatase B, this enzyme is deficient. So we have a clinical and a biochemical diagnosis of mucopolysaccharidosis type 6. This is the first level of investigation. That brings us to the next level, that is molecular characterization, because we want to find the mutations. So sequencing here shows that he is homozygous for this particular mutation D53N, and it is a novel mutation. Now this creates a dilemma. We have to know whether this is really the mutation or not. So how do you prove it? Because it has not been reported till now, and so we use various strategies. The first strategy will be to use various prediction software. So these are softwares which tell us whether it is likely to be disease causing or not. But the softwares also can make mistake, but we use them as the first line to tell whether it is disease causing. So this software is telling that this particular mutation, all of them are telling that it is likely to be disease causing. We go to the next step. We try to model this protein. So we make a protein model using earlier protein, normal protein model, and introduce this mutation and see whether it changes the interaction with other molecules. So this is the next level of evidence that we gather, that is protein structure characterization. And then the third level is functional characterization, where we take out mRNA, make cDNA, and put this cDNA into the cell, or create the mutation and then put the cDNA, and then look for what is the change happening with the mutation and without the mutation. That gives us further confidence that yes, this is likely to be the mutation. As we can see here, this is without the cDNA. When we put the cDNA, it goes up 10 times higher. This is with another polymorphism, and with our mutation it comes down, which almost proves that this is a disease causing mutation. We can give the report to the family. But this may not always be true, because in science we try to find more and more evidence, and this evidence is only at the cell line level. This evidence is not at organism level. So always in science we have to say dil mange more. We want more evidence. We want more to be sure that it is the real variant, and that is where the model organisms come. So you could use either a worm model or a Drosophila fly model or a fish model, zebrafish, mouse model, or a chimpanzee model. That would be used, and you see whether the same phenotype occurs in the model organism also. Or nowadays you have another method called as induced pluripotent stem cells, where if a patient has a brain disorder, you can't take his brain cells to study. So what we can do is we can take the cells from his body and then convert them into pluripotent cells and then convert them into neurons. Now although we don't have the brain cells from the patient, we have the neurons which are almost similar to the brain cells of the patient, and then whatever new treatment that we are developing, we can study on this instead of studying it directly on the brain of the patient. Similarly, you can see this is a model made from the cardiac cells, and then when the model of cardiac cells is made, they start beating because they have now become cardiac cell, they do their function. So in this way we have different models that are used, and ultimately at the end of all this we are able to give the family that दूध का दूध पानी का पानी हो गया. This is the mutation which has caused the disease in your family. Now you use it for doing the prenatal diagnosis, or when you find a new gene, you develop treatment towards it, and that is what is the next letter C. So what does it matter to the family if you found a variant or a gene? It doesn't mean anything. They need care and cure, because every disease needs cure. You want to cure the patient. So the medical paradigm that we have been doing till now, or what is mostly practiced, is that a sick patient comes and then the doctor goes through the clinical features and gives some treatment and the patient is happy. But in genetic counseling, either a sick couple or a sick patient comes, but we don't prescribe medicines. Mostly we prescribe words. That is what medical geneticists do till now. So genetic counseling is what we do, where we tell the family about the details of disease by doing the diagnosis, the cause of disease, whether any treatment options are available. It is very important to tell them if treatment is not available. That is also very important. Otherwise they go doctor hopping here and there. We tell the inheritance pattern, the risk of recurrence, whether prenatal diagnosis can be done, and ultimately we hope that a happy family can be formed. However, there are lots of general misconceptions, and this Dr. S.S. Agarwal many times used to point out that people feel genetic diseases are rare, or genetic diseases are always inherited, family history is a must. And this was his very favorite thing. Any doctor would come to him and tell that genetic disorders are not treatable. He would tell, diabetes ko treat kar sakte ho, hypertension ko treat kar sakte ho, then don't speak. So we are trying to control disease. We are not God. We cannot cure everything. But still there is hope for family with genetic diseases. And nowadays we should say even better, because newer therapies are coming for genetic diseases. So not only diagnosing genetic disorders, we are able to treat many of the genetic disorders using various treatment strategies like replacement of deficient protein, enzyme replacement therapy given for various lysosomal storage disorders. We can restrict the harmful metabolite like phenylketonuria or MSUD. We can give deficient diets to the patients. We can transplant the bone marrow, liver or kidney if the disease is causing abnormality in that organ. We can do chelation therapy in Wilson's disease, targeted therapies for cancer, and supportive therapy, genetic counseling is always there. So most of the therapies now are basically limited to substrate limitation, product replacement and supportive therapy. But newer and newer therapies are coming up as we know more and more about the diseases. And if we look at different levels, so we have treatments at different levels. So at the gene level, you could do gene therapy, which is still in research stage. At the mutant RNA, you could do RNA interference based therapies. At the mutant protein, you can replace a normal protein. At metabolic function, you can do dietary therapy or pharmacological interventions. At the clinical phenotype, if there is a polydactyly, remove that finger. You saw that patient, the abnormal fingers were treated by surgery. And for the family, we can do genetic counseling, prenatal diagnosis. So it's not as if like we really cannot treat these disorders. But treatment is changing, and that brings us to the last letter T. And this genetic information is really now turning our modern medicine towards traditional medicine. So what the ancient Ayurvedic medicine or the traditional medicine, what would happen is that a typical patient will go to a Vaid, he will do some of his investigations, Nadi Pariksha, he will make his own medicine for that patient only, not a factory based medicine, and then give patient that medicine along with diet, lifestyle and the medication. So this is a holistic kind of treatment that was followed, as opposed to the modern medicine where doctor sees more of the reports, more of the investigations, and then all patients with fever are given paracetamol, all patients with malaria are given chloroquine, all patients with one particular disease. So we have these silos of different treatment for different diseases. But slowly genetic knowledge is changing this towards something called as personalized medicine. So this current practice of trial and error of one size fits all is changing to the right treatment to the right person at the right time, and that is what is the future of medical genetics or the future of medicine as a whole. And this was the favorite example of Dr. Agarwal, which he used to tell always to explain about personalized medicine. So 6-mercaptopurine is the drug which is commonly used in treating acute lymphoblastic leukemia. Now if you give 100 patients this particular drug, there is an enzyme in our body, thiopurine methyltransferase, which metabolizes this. Now what happens is that out of this 88% of the patients, they have normal enzyme activity, they will have relapse of the disease. The disease will get cured or treated. But 11% of the people have intermediate activity. So these yellow shaded people, they have low activity of this enzyme. That means they will have lots of side effects. And 0.3% or 1% here will have undetectable activity of this enzyme, and you give the drug and he will die. Now if you could do some test and identify this 1% and these 11 persons, you could give low dose treatment to these 11 and don't give the drug to this fellow, and all 100 would be happy. And that is what is personalized treatment. You first do a test, find the genetic makeup of the patient and then give the treatment to him, rather than doing a trial and error. And this is known for multiple different diseases, as we do in our routine clinical practice also. All patients don't respond to a particular drug. So hypertensive drug, about 10 to 30% will respond. So all different drugs, people have seen that different percentage of people will respond to these drugs. So what is present is the reactive medical therapy, where there is diagnosis followed by one drug is given, that is not working, you switch the drug, then switch again, and the patient is sick for a long time. Instead of that, personalized medicine is that you diagnose the disease, give the right drug, and immediately the patient gets treated. So his long time of disease is avoided. And even better still is predictive medicine, where if you can find predisposition, you can find first only that he is predisposed to a particular disease. If you put some preventive measures, then even the disease will not occur, and that is the ultimate medicine that could be practiced. That is where the whole world is moving towards. And this is not really in theory. Already there are various tests that are available for thiopurine, TPMT, polymorphism analysis. We are actually doing many of these tests now. Also in cancer, there are targeted therapies for many of the disorders where without doing a test you can't give treatment for that particular cancer. So this is already becoming a reality, and in future probably when the baby is born, you take a blood, sequence all the genes, get to know all the proteins, all the metabolites, store it in something like a credit card, and every time a patient is sick, he just goes with this card, all the information is there, and you know which drug needs to be given to that family. So instead of making a kundali with all the Rahu Ketus, we'll make a microarray kundali, a genetic horoscope, and that should make all the patients happy. So thank you all. I have very sweet memories of this place. The 3 years I spent here have been really the great years of my life. I thank madam and thank you all for patient hearing. Thank you.